Okay, so again for the cell cycle, here you have a cell in interphase, which is made of G1, and then you can see the cell is preparing for division. It's on a timer, so here we go again. And you can see here as it gets into S phase, the DNA duplicates, and you get a second set of DNA. And so at this point, our cell has grown, it's prepared for division, it's doubled its organelles, it's made centrosomes, and it's going to prepare for M phase. So here, at the beginning of M phase, the chromosomes, they actually condense down and become visible. And then you have these things called centrosomes, and the centrosomes is where the spindle fibers are going to come from. Here you have the nuclear envelope breakdown, and then you have the centrosomes are going to move to opposite sides of the cell. They're going to send out things called spindle fibers. The spindle fibers attach to the centromeres, and the centromeres are what's holding the two sister chromatids together. So here as the spindle fibers, they start to pull the chromosomes towards the centromere. The centrosome. However, the other centrosome also sends out spindle fibers, and you can see them attached to the centromeres. And it results in a tug of war that ends up with the chromosomes that are duplicated, made of two sister chromatids, along the middle of the cell. And we're going to, actually they will separate and get pulled apart to opposite sides of the cell. And then you have new nuclear envelopes will form, and the chromosomes are going to uncondense and go back to like chromatin. At this point, mitosis is technically finished because the nucleus has divided. And then our cytoplasm is going to begin to pinch in. This area right here is called a, let's see if we can go back. This area here is called a cleavage furrow. So right here is a cleavage furrow, and it begins to fold in. And now you have two daughter cells. Okay. See if you can try this out. Hit pause real fast. All right, so when we look at the relative amounts of DNA in G1, it's when we start out at this much. Then in S phase, it duplicates. Then G2, it's still duplicated. And then in M phase, that's when the sister chromatids separate. And now we're back at G1. Awesome. So now let's go. Oh, here you can kind of see the cleavage furrow. So that's the cytoskeleton folding in. If you've ever changed a baby's diaper, it also looks like a baby butt. But whatever. So now when we look at the regulation of the cell cycle and cell division. So we need to, when our cells divide, they need to coordinate all of these events that are happening and the rates that the cells are dividing. And so it needs to happen. If you think about humans, we have 200 different cell types and different kinds of tissues. So the rates of division are going to be different. For example, your taste buds, you get new taste buds every 3 days or so. Those are specialized cells that help you taste things. And so when you burn your tongue, it's not forever because those cells eventually fall out. You swallow them, and then you have new ones replacing them by mitosis. Your blood cells last about 4 months. You get new skin cells every 24 hours. The walls of your stomach, because of that stomach acid, are constantly being replaced by mitosis. And then you have some cells that don't divide very often, like a brain cell or a liver cell or a muscle cell. Some cells just don't divide. So the cell cycle is going to be different for all different kinds of cells, and it needs to be coordinated and happening at the appropriate times. So when we look at our cells, here's just some more words about what I just said. Okay, so when we look at our cell cycle, though, it is regulated by different checkpoints. So with this, we have stop and go signals that can be both external, meaning they're from outside of the cell. So imagine or remember like protein receptors, like a tyrosine kinase, for example, that's going to receive like a growth factor or a signal coming in from outside saying, hey, time to divide. Or it can also have internal signals, like when do we go from G1 to S? How about from S to G2 or from G2 into M phase? When is the cell ready? When does it go on through that cell cycle? So there are things called checkpoints within the cell. And so if we think about this, in the G1 checkpoint, we're going to make sure that all of the DNA is healthy. So in that G1 checkpoint, can DNA synthesis begin? Are the chromosomes all intact? Are these good, healthy cells? And if they're not, the cell will receive a message for cell death. So like apoptosis, which is programmed cell death, well, now it'll receive a signal and then it'll die and not reproduce. Because why would we want to reproduce unhealthy cells? This is also the step, though, where if there are errors in the DNA or in the cell, sometimes it can like run this stop sign and then progress through the cell cycle when it shouldn't, and that's uncontrolled cell growth, which is like cancer. So there's a checkpoint, though, right between G1 and S to make sure that the DNA is all good and ready to go for division. Now, between the G2 and M phase, was the DNA finished copying? Do you have sister chromatids? All 46 of our chromosomes duplicated? Because if they're not duplicated, now you're going to end up with daughter cells that aren't identical, that don't have exact copies of the DNA. So at this point, you have a commitment to mitosis. They're going to divide if they pass that G2 checkpoint into M phase. And then you have your spindle checkpoint. So with this, are all chromosomes attached to spindle fibers? And did they separate correctly? We'll learn in meiosis how sometimes there can be errors, and the sister chromatids, instead of separating, they stay together and end up in a daughter cell. Now that daughter cell has an extra chromosome or a missing chromosome, which we can see, like a common example is in Down syndrome. Down syndrome, they have 3 chromosome 21s, and so that could be a result of chromosomes not separating correctly when gametes, eggs, and sperm are being made. Right now, cell cycle, we're talking a lot about somatic cells, our skin cells, but we can see mistakes of chromosomes separating big time when we talk about sperm and egg, because now we see it in our offspring. Okay. All right, so those are our checkpoints. And here's just a slide. You can like pause on it and read it if you'd like to. All right, so now the G2 checkpoint, the most critical checkpoint, is between G1 and S phase. Now this is like a stop sign to say go. So this checkpoint is really like checking, is the cell good? Is it ready for division? And if a cell receives a like go signal, it's going to divide. If a cell passes the G1 checkpoint, it's going to divide. And now there can be 2 types of signals about whether or not this cell will go into S phase, and it can be internal signals or external signals. If a cell does not receive a signal, it will enter into that G0 phase, which is basically like a not dividing phase. And so really the whole cell cycle all depends on signals. That's why I have this here. It's going to be either go or stop signals. So in the G0 phase, this is the non-dividing phase, and a lot of our cells that are not in active mitosis are in G0. They're living their life, making their proteins, doing their jobs. But now, how do cells know when to divide, right? So with this, they're going to communicate with signals. So our signals are chemicals, and they give the cell clues about their environment. And when we talk about signals, a lot of times we're talking about proteins. We can have inhibitors or we can have activators. Now, one of the first cool experiments that was done where scientists actually figured out that there's internal chemicals that tell a cell when to divide or not is they took a cell that was in M phase. So it's dividing, the chromosomes are getting separated, the cytoskeleton's getting rearranged, and they put it right next to a cell in G1 so that their cell membranes like fused. Kind of like if you put 2 bubbles together, how they like can form one. So now those chemical signals that were inside, well, they're going to be mostly proteins, inside of this M phase cell, like diffused into this region and activated this cell to go into M phase and divide. Even though, check out its DNA. Its DNA hadn't duplicated. It skipped S phase and G2, but it still became an active dividing cell. And that was one of the first signs that, wow, there must be some chemical signals within the cell, internal molecules that tell a cell to divide or not. Okay, so let's go ahead and talk about these signals. So there are protein signals that promote cell growth and division. So growth factors are an example, and we can see here you have a growth factor attaching to a receptor protein. Now you have your signal transduction pathway that will control the cell insides, telling it to divide or not divide. So here you have your growth factor, you have your protein kinase, your phosphorylation cascade. Then you have some, we're going to learn about CDKs. You have some kinases that become active inside the cell that lead it towards cell division. So here we're going to look at an example called platelet-derived growth factor. Now, when you get cut and you're bleeding, and then eventually it starts to clot together and you're not bleeding anymore and you have the beginning parts of a scab, those little tiny pieces that are starting to form that scab are actually your platelets, which are like pieces of red blood cells, if you want to think about it that way. So let's really think about this big picture. You just got cut. Now you have blood coming out. You have an injury. And your mitosis, like your skin cells, your somatic cells, need to heal and replace and grow back together. So here, when you've been cut, you have platelets coming to the area to stop you from bleeding to death, basically, and to also stop bacteria from getting inside, to repair that barrier of your skin. So now your platelets are going to release a growth factor. They're going to release a chemical signal saying, hey, there's been a break in our skin. There's a breach in our protection. Our skin is one of the most important organs in our body, one of our most important parts of our first line of defense of our immune system. It keeps things out. And so if there's a damage to it, now pathogens can get inside. So it makes perfect sense that the first responders, your platelets, that are going to block bacteria from entering and blood from leaving, are going to release chemicals that say, hey, time for cell division. We need to repair this cut as soon as possible. So now when we think about how this works, PDGF, platelet-derived growth factor, when it attaches to a nearby cell, so the platelets, the little pieces of blood cells, are releasing this growth factor. So when it attaches to nearby cells, that's going to activate those cells to now divide, because once they divide, they're going to start to multiply, and eventually they'll come in contact, creating that new skin, repairing the wound. And so here you have your signal transduction pathway, and that final cell response is cell division. It's going to activate that nuclear envelope breaking down. The chromosomes are going to condense. It's going to go through S phase, G2, and M phase and result in two identical daughter cells. So when we look at these growth factors, there's these internal signals. You have external signals, which are your growth factors that attach to protein receptors. And then you have internal signals that will say, hey, it's time to go from S phase into G2 or from G2 into M phase. So what moves the cell on the inside from G2 to M, for example, are promoting factors, these internal signals. And these internal signals are kinases. So let's go ahead and see. And this is the primary mechanism of control for cells moving through the cell cycle. And if you remember kinases, they are responsible for adding phosphate groups for phosphorylation and activating proteins. So let's go ahead and see how this works. Inside the cell, we have cyclin-dependent kinases, CDKs. So it's cyclin-dependent, which means it needs a cyclin to be active. So here, the cyclin-dependent kinases, these are always in the cell, but cyclin is what changes throughout the cell cycle. And it's going to help to regulate the proteins there. So let me be more clear. You have these cyclin-dependent kinases that depend on cyclin. When cyclin is there and it attaches to the kinase, now that kinase is active. And it can help move that cell through the cell's checkpoints. And so the cyclin-dependent kinase is what helps move the cell from like G2 into M phase.